In this video, I'll show you two examples of how the environment can alter a phenotype of an organism. You've likely learned that a person's genotype dictates their phenotype. Which alleles they have for a gene will result in the production of a specific protein, and the function of that protein creates the person's phenotype. But that's not the whole story. It turns out that the environment can alter your phenotype without altering your genotype. This can happen in two ways. Your environment can trigger or shut down the expression of a gene. Alternatively, your environment can alter the activity of the protein created through gene expression. In both of these cases, this means a different environment can cause two cells with the same genotype to have completely different phenotypes. Let's look at this scenario where an environmental factor can promote gene expression. An example of environment triggering gene expression is seen in the E. coli lac operon. This operon contains a group of structural genes that encodes proteins necessary to break down lactose. The E. coli genome also contains a gene that encodes a repressor that will bind to the operator of the operon, turning off the expression of the operon structural genes. When the repressor is created, it's in an active form, so it can bind to the operator of the operon and prevent the expression of the operon genes. When lactose is added to the environment around the cell, it enters the cell and binds to the repressor, making the repressor inactive. When the repressor is inactive, it cannot bind to the operator, so the operon can more easily express these genes. From these genes, proteins are produced to help metabolize lactose, and when the lactose is no longer present in the environment, the repressor proteins can resume their active state and prevent further expression of the operon genes. In this case, the absence or presence of the environmental factor lactose dictates whether or not the genes of the lac operon are expressed. When lactose is present in the environment, the structural genes are expressed, and the absence of lactose will stop the genes from being expressed. There you have it. Adding or removing lactose from E. coli's environment can dictate whether or not it expresses the genes of the lac operon. The environment is altering gene expression and therefore what proteins the cell contains. Now let's look at this scenario, when the environment alters the activity of the protein produced from the gene. An example of this can be seen with cat fur color. Cat fur color is partially controlled by a gene that encodes an enzyme that catalyzes the production of the pigment melanin. This melanin gives the cat darker fur. Now there are two alleles we'll focus on in this example. The first, let's call it FA, encodes an enzyme that catalyzes the production of melanin. The second, let's call it FB, encodes a version of that same enzyme, but it differs in that it is temperature sensitive. At normal core body temperature, the enzyme is non-functional, meaning it won't be able to help produce melanin. At temperatures lower than body temperature, the enzyme is functional. So the activity of this enzyme is environment dependent, temperature dependent. Siamese cats are homozygous with two copies of the FB allele. So their cells are only making this temperature-sensitive version of the melanin-producing enzyme. So in the core of the body, where it maintains a high core body temperature, the cat's fur will lack melanin and be this lighter color. But at the cat's extremities, where the temperature may be lower, like its nose, ears, feet, and tail, the enzyme is functional, allowing for the production of melanin in the fur in those areas. This temperature sensitivity gives the Siamese cats their signature fur color pattern, with darker areas around the nose, ears, feet, and tail, where the enzyme is able to make melanin, and a lighter body where the enzyme is not able to function to make melanin. Despite each of the body cells in the Siamese cat having the same genotype and producing the same enzyme, in some locations the enzyme works, and in some locations the enzyme does not. So you see different phenotypes developing across the cells of the cat based on an environmental factor, temperature. It is the presence of the FB allele, which produces the environmentally sensitive enzyme, that gives the environment the potential to create different fur colors across different areas of the cat's body. Alternatively, a cat that is homozygous for the FA allele would be making the non-temperature sensitive version of the enzyme and would be able to produce melanin and create dark fur all over the cat's body. In this cat, the difference in temperature between the body and the ears or nose or tail does not alter enzyme activity. So, in this case, a homozygous FA genotype does not have the potential for environmental temperature to alter the cat's fur color phenotype. These examples are just a taste of how the environment can alter phenotype. If you'd like to learn more, check out the resources in the video description.